Hey guys, this is Gabby and Sierra on our project about energy expenditure and foraging behavior of the California sea otter.
The California sea otter is found along the west coast of the U.S., and the species is called Anhydra lutris neris. They eat urchins, clams, shellfish, snails, mussels, and abalone.
They require an average of 8,600 calories a day and weigh up to 36 kilograms per individual otter. They dive meters under the sea for up to 2 to 5 minutes to forage for food. They have a high metabolic rate because of their small body size. This requires them to eat 25% of their body weight each day. They have evolved to use tools to eat their food, such as rocks and other materials found in the sea.
We studied foraging, dive, and feeding behavior in sea otters. We hypothesized that the longer the dive, the bigger the food, and therefore the longer the surface time. We hypothesized that otters would prefer to dive for the least amount of time to acquire the largest prey.
We studied these otters at the Wharf and Elkhorn Slough in California. We timed otters diving and surfacing while taking observations on the food size, if they used tools, and if they groomed. We collected data on 20 otters, focusing on one individual feeding at a time. None of the otters we observed had pups, and gender was not taken into account when we observed their times of diving and surfacing.
For the data analysis, we first did a multiple regression of dive time to food size and tool use. This was not significant. Second, we did a multiple regression of surface time to food size and tool use. This was significant for food size with a p-value of 0.005. This means that tool use did not affect feeding or dive times, but food size affected the surface time. This is because larger the food item, the more time it takes to eat.
Next, for our results, we did a chi-square test comparing food size to tool use. This shows how often small or large prey requires a use of a rock to break it open to feed. It was significant with a p-value of 0.0334. This shows that some large prey required a tool to be opened, but no small prey required a tool.
Next, we did a linear regression of dive time to surface time. That test was not significant. We then did a multiple regression of dive time to surface time and food size, and then a multiple regression of surface time to dive time and food size.
The first regression we did was significant for both surface time and food size. Surface time's p-value was 0.0419, and the p-value for food size was 0.0071. This showed that dive time is affected by surface time and food size.
The second regression test was significant for both dive time and food size. The p-value for dive time was 0.0419, and the p-value for food size was less than 0.0001. These results show that surface time is longer when the dive is longer. This could be from the need to recuperate and groom after a long dive.
The last test we did for our results was an ANCOVA test, and we did a no-intercept model and an intercept model. For the no-intercept model, it was not significant for food time to food size times dive time. The intercept model was significant for surface time to food size times dive time. This has a p-value of less than 0.0001. This means, once again, that food size and dive time affects surface time. We did the test for the intercept model because it is a better representation of biological interactions. There are trade-offs between diving to obtain the food and foraging the most energetically beneficial prey. Other studies have shown that dive time and surface time are most affected by prey type and prey size. This helps us conclude that otters will behave in the most cost-effective foraging strategies for gaining food. They will attempt to gain as much energy and calories while keeping a short dive time. Each individual specializes in a certain food type and size to eat efficiently and dive efficiently. This specialization is more advantageous when food availability and food type varies between each environment that they forage in. There are many possible future studies. Does foraging behavior differ between age, environments, sexes, female with pups? These are more questions we could answer with behavioral studies on their foraging by taking into account gender, age, and resource availability while observing dive and surface times out in the field.